Okay, this is General Biology, Bio 131, and this is Chapter 8. So, Chapter 8, let me share the slides. Chapter 8 is about the microscopic organisms. So, microscopic organisms, these are the very tiny organisms that we can see using the microscope, hence the name microscopic organisms. We will go one by one from the origin of life to what prokaryotes and what fall under this with the different categories and eukaryotes, and then the rest of the organisms. So we start with the prokaryotes. If you know what are the prokaryotes, and eukaryotes. If you don't know, this is what we're doing. And simply, the first thing that we need to know is what's pro and what's karyote and what's eu, which is EU. Pro means before. Karyote means the colored, referring to the nucleus. So prokaryotes, these are the organisms that does not have a nucleus because it is prokaryote. It was hundreds and thousands of years before it's advanced enough to have a nucleus. It still have genetic material, but it's not in the nucleus. So this is called prokaryotic. Eukaryotic, on the other hand, UEU is a prefix. It means normal. So eukaryotic or karyote means the color referring to the nucleus. So these are the ones that does have a nucleus. So it's very important to know what eukaryotes and what prokaryotes. So eukaryotes, which are the ones that have a well-defined nucleus with nuclear membrane. So the genetic material is located within an actual nucleus. So that's why we call it eukaryotes. EU, normal, karyote is the nucleus. So this is the one that have normal nucleus. Prokaryotes, pro before, again, pro before. It was years before it's advanced enough to have the genetic material within an actual nucleus with a nuclear membrane and so on. So under the eukaryotes, there are different organisms that are considered eukaryotes. Obviously, all of them will have the minimum. This is not the only thing. The minimum is well-defined nucleus within a nuclear membrane containing genetic material. This is the minimum. So now you call it eukaryotic. We will have more details. But which ones are the subcategories like eukaryotic, including what? Including plants, animals, and fungi. Plants, animals, fungi, including human being, of course, being classified in biology as with the same category with animals. Prokaryotes, we will learn the subcategories. So the prokaryotes, these are small, very small one, simple. Obviously, this is more ancient. This is more older, billions of years ago. This is what you used to have before to start to advance and develop a nucleus in which we have the genetic material included within a membrane and so on. So the prokaryotes, this was millions, billions, I mean billions of years ago on Earth, and it's very small and simple. Later on, it started to advance. So when we talk about the prokaryotes, which is so far no nucleus, but it doesn't mean that they don't have genetic material. They do have genetic material. If you don't have genetic material, it's not considered an organism in the first place. It's not considered a cell in the first place. You have to have it. Everything have it. But where do you have it? You have it within a nucleus, eukaryote, plant, animal, fungi, or you just have some genetic material not well defined, not in the nucleus, and so on. That will be eukaryotes. So obviously, prokaryotes also have genetic material without a nucleus. Subcategories or the domains that fall under Prokaryotes are domain bacteria and domain archaea. So eukaryotes, what are the domains under the eukaryotes? EU, normal. This is plants, animals, fungi. These are the three domains under eukaryotes. Now we're talking about prokaryotes. You have bacteria, you have archaea. Both of them are unicellular. It's one cell by itself. When we discuss the other ones, it can be uni or multi, but this is unicellular. Uni means one. The bacteria can be found everywhere on Earth, on humans, animals, plants, on earth, on lands, on water, anywhere it can be found. Some of them are bad, some of them are good, some of them are beneficial, some of them are pathogenic or can cause disease. So not all bacteria are bad. Actually, there are bacteria in our gut, in our colon, that's good enough that without it, you can have bleeding disorders. It makes vitamin K and other things. So bacteria can be good or bad. It's everywhere. Archaea, on the other hand, is also unicellular, just like the bacteria. Bacteria is one cell. Archaea, one cell. But bacteria is everywhere. Archaea is not. Archaea, most of the time, it can be, but most of the time, it's in extreme conditions. Extremely cold, extremely hot, extremely humid, and so on and so forth. What else is in common between the prokaryotes, including the two domains, bacteria and archaea, besides that they have the genetic material without a nucleus? Beside also being small, simple, and one cell. What else? They all have something called cell wall. In the when we go to eukaryotes, this is not the case. Some and some. What's a cell wall? Is that just what surrounds everything in that cell? No, it is not. What surrounds everything in the cell is called the cell membrane, not wall. Membrane. Every single cell on Earth, eukaryote, prokaryote, whatever it is, you have to have a nucleus. Around it, you have to have a cytoplasm bounded by a membrane. This is to be cell in the first place, whether eukaryotes or prokaryotes, it doesn't matter. Otherwise, it's not a cell in the first place, so it's nowhere around what we're talking about right now. So when we're talking about it's a cell, it's an organism in the first place. To say it's an organism, you have to have genetic material in general. Genetic material, you have to have a cytoplasm, and you have to have a membrane, basics. Now the categories again, eukaryotes, prokaryotes. This depends on nucleus is number one and other things. Do you have a well-defined nucleus with a membrane containing genetic material? Eukaryotes. You have genetic material still, and obviously cytoplasm and a membrane. So, but not within a nucleus, you don't have a nucleus. Before development of the nucleus, you don't have nucleus, but you still have the genetic material, cytoplasm, cell membrane, not a wall, it's different. So this is a prokaryote. So if it is a eukaryote, you have the genetic material inside the nucleus, surrounded by a nuclear membrane. You have a cytoplasm, you have cell membrane. Prokaryote, you have genetic material, not in the nucleus, but you do have cytoplasm and cell membrane plus a cell wall. So the cell wall is an additional thing, like you're wearing a shirt like this plus a jacket, or not. In this case, all prokaryotes, bacteria, archaea, they all have a cell wall, which is an additional layer to provide extra protection. So this is all of them. Flagella, not all of them. Some of them will have flagella. Flagella is like a tail that when it moves, it helps these organisms to move, propelling the cells. Capsule, some of them have the capsule, some of them does not have a capsule. What's a capsule? It's even more additional layer. So you have the genetic material in a cytoplasm. and then you have the cytoplasm, and then you have a membrane. Outside of the membrane, in prokaryotes, all of them, you have a cell wall. Outside of the cell wall, some of them will have the capsule, and some does not have a capsule. So what you have to have in the prokaryotes? Genetic material, not in the nucleus, definitely, cytoplasm, cell membrane, plus cell wall, plus or minus flagella and capsule. What else in relation to the prokaryotes? Some of them, obviously, remember when I say all of them, different than some of them. It makes a huge difference. All of them, genetic material, all of them, cytoplasm, all of them, cell membrane, all of them, cell wall outside the cell membrane. Plus or minus what? Capsule, maybe yes, maybe no. This is something additional to make them stick so that they stay in place, they're not washed away. It's extra protection. The capsule is extra protection, but it's plus or minus. Flagella, plus or minus. Endospores, plus or minus. Some of them will have endospores and some of them does not have endospores. What's the endospores? Endospores is simply if this is like a bacteria or something, and the condition is too harsh for them to live their normal life. So what they are going to do, they are going to shrink, become smaller, have a thicker wall. They become smaller, have a thicker wall, and they kind of dormant. So they stay like this until the condition improve, and then they are going to return back to the normal status. But what they do temporarily is they shrink and they add harder, thicker wall outside. So now they are too small, so that whatever the nutrients that they have, it's okay. They can help them to stay alive. They don't die, and that's why they are too small. Just the minimum, just like the plants. Look at the plants. If you have a tree or something, it's like in the winter, all the fruits disappear. If it's a fruit tree, for example, all the leaves disappear, everything disappear, and you see just looks like a stick. Is it dead? It's not dead, but it's shrinking and just keeping the inside alive so that when the condition improve in the next spring or something, they are going to flourish and restore. But they didn't die. They just go into that phase temporarily, which is here it's called the endospores. So they shrink, apply a thick wall to protect them from the harsh conditions until the condition improve, and then they become bigger and the cytoplasm become bigger, and that's protective membrane or wall becomes just a normal one, and so on. Um, Prokaryotes also usually they reproduce by binary fission. Like if you ask, like this is one bacteria become 2, 4, 8. How do this is reproduction? How do they reproduce? Like male and female? No, it is not. They split into halves. So here is 1, become 2, and each one of those split into 2 that become 4, and so on. We call that binary fission, and this is very important to remember, splitting into 2, and each one of the 2 will split into 2, that will be 4, each one of the 4 will become 8, 16, 32, 64, and so on. And because of doing that, it's very fast, so they can fuel bacteria 10, 20, 40, and so on, 80, 160. That will happen in very fast pace, so they can go into millions in a short period of time. Regarding the shape, prokaryotes can be in a form of spherical structure, like a ball, and we call that coccus. The plural is cocci. And this is something, by the way, in Greek and Latin, since you're studying biology, you need to know basics. These are basics like prefix and suffix and how to differentiate between single and plural and so on. So usually in Greek and Latin, which is the language of biology and then anatomy and physiology, this is the basic. language. So you need to understand that. Usually the difference between single and plural is it US at the end or I? So caucus or caucai. Caucai, this is the plural. So if it looks like this, spherical structure, we call it caucai. If it looks like this, rod shaped, we call it bacillus. If it looks like spiral or curved like this, we call it spiral shaped. Remember um, that they are individual cells. They can come together, but still considered one cell each. So this is the shape. Spherical structure is caucus. Rod-shaped, bacillus. Spiral or curved, we call it the spiral prokaryotes. Um, in order for these prokaryotes to survive, they will have different nutritional habits. They feed themselves or they eat in a different way. So if you think about it, you would say prokaryotes like bacteria and archaea, of course they eat something. They invade like our body. No, no, it is wider than that. It's more than that. This is what we know. But actually it's more than that. This is one type. They consume other organisms, like they consume our nutrients from our body. They go to like invade animals and they take the nutrients from the animals. This is one type, not all of them. Some of them, very few, they can do photosynthesis. You think about it, photosynthesis isn't photosynthesis like the plants do photosynthesis. They take energy from sunlight and they convert it together with water and carbon dioxide into nutrients. Actually, yes, some of the prokaryotes are able to do the same thing. They can do just like the plants. So this is photosynthesis. Some of them, like those weird types of prokaryotes that live like very deep in the water and in like in the ocean or under the like very deep in the oceans. You think like how do they survive? Photosynthesis. Where did they get the sun from or the carbon dioxide from? And this is even a salted water. So how can they get their nutrition? This is an additional thing. Like these, in this case, um, they can take uh, their needs from the surrounding sulfur-rich superheated water. The water is superheated, and they can actually get their nutrients from that environment around them. Um, Prokaryotes usually form something called a biofilm. A biofilm. They are not like scattered like this. They usually make, not 100% of the time, but vast majority, most of the time, they make a biofilm, which is colonies that organized in a specific shape, and they come together and make this biofilm, like that biofilm, like in this picture, around your teeth. This is not like one here and one there. This is a whole film of these prokaryotes. This can happen on our teeth. This can happen on the plants. This can happen on the rocks. But most of the time, they do a biofilm. They make a film, not scattered. Most of the time. Um, Now, what if these organisms they live in extreme conditions? Extreme conditions is like too hot, too cold, too salty, like any extreme. How do they survive? Can they still survive? They can, depending on which type are we talking about. Um, and this is specifically which type of prokaryotes we're talking about. Archaea. Archaea. Specifically Archaea, they can survive unlike bacteria. Bacteria, most of them, they survive in the normal environment. But most of the Archaea, which is another domain of the prokaryotes, they can actually survive and live their normal life in extreme conditions, in extreme habitats. Like some of them are called methanogens. These are the ones that produce methane gas. We will talk about it. Halophils. Halo means salt. Phils, like philic, like phobic versus philic. Hydrophobic, hydrophilic. Hydrophobic, you hate water. Hydrophilic, you like water. Halophils, these are the salt lovers. Remember, philia is the opposite of phobia. Philia, phobia. Philic, phobic. You like, you hate. Philia, like. Phobia, hate. So halophils, halo means salt, phils means lovers. They love the salt, and this is normal habitat for them. Thermophils, these are the heat lovers. Like in some areas that's extremely hot, it's okay for them. This is their normal habitat, and they like it, and this is where they survive. Some areas contain like caustic chemicals, like some of these in Africa and in different areas in the world. Uh, they they have this caustic, literally caustic, because it's extremely salty, and some archaea they just like it, and they are totally fine with that. The halophils. Uh, methanogens. Methanogens. You produce methane gas. Aren't you supposed to take oxygen and produce carbon dioxide, or take carbon dioxide and produce oxygen like plants? No, they don't do that. This type, they actually produce methane gas. Their waste products, like us and animals, we take oxygen, and our waste product is carbon dioxide. And them, the carbon, it's not a carbon dioxide, it's methane gas. They they produce methane gas, and this is usually located in a thick mud, like at the bottom of a swamp or bog or something. At the bottom, it's very muddy, thick mud, and over there is no oxygen, and this is what they like. Uh, uh, that methane gas actually, they can put like a well or something and collect that methane gas coming from decayed garbage or anything that's decayed, and they can have some uses for it. But what did that is called the methanogens. They take whatever they can find. No oxygen, no oxygen. They do not take oxygen and produce carbon dioxide. No, they are unique. They take whatever surrounding them deep in this environment, and they end up producing methane, unique. So they can put a well and collect that methane and use it for different industrial needs. And by the way, those that they don't like oxygen, they don't need oxygen, they don't want oxygen, they don't like oxygen, they know how to get the nutrients from oxygen-free environments, like all the way at the bottom of like a swamp or something. They live there, and there is no oxygen, and this is what they like actually. And because of that, they call it anaerobe. An is a prefix that means no or without. Aerobe is coming from air, and they actually mean oxygen. Anaerobe, no oxygen. Anaerobic, this is what it means. Next one is called the halophils, and we just mentioned that they live and they like halophils, salty environment. Uh, they can survive in an extremely salty environment. It can be like 10 times more salty than the seawater. Like you see the seawater, like if you taste it, you say that this is salty, very salty. It's 10 times. So it's extremely concentrated, like the Dead Sea have something like this. And that's why the Dead Sea is called dead, because no other organisms can survive there. So everything is dead except for those. And maybe similar organisms that can actually survive that. The next one is called the thermophils. And again, phil means like. Philia versus phobia. Philia, phil. So thermo means heat. Thermophils, the heat lovers. They live in an environment that have a very high temperature, above boiling, above the 100 degrees centigrade, which is the boiling for water, for example. It's above that. So nothing else can survive beside those thermophils. Um, and this obviously was uh, somewhere around like volcanoes and extremely hot areas, which is the very ancient types of these prokaryotes or archaea in this case, that this is their environment. They live there. So So the bacteria, which is one of the two types of the prokaryotes, um, this is huge amount of bacteria, too tiny. So compared to our cells, but there are millions of them, including in our body. It can be like in the intestine or something. Um, and some of them are beneficial and some of them are harmful. And we discussed that. They live like everywhere on Earth. And again, some of them can be harmful, some of them can be unharmful, and some of them are actually can be helpful. Um, this again, we're talking in this chapter, we're talking about microscopic organisms. So these are all tiny organisms that we regarding the cells, their cells, that we can see using the microscope. So if we're talking about the bacteria, we said that the bacteria can be harmful, can be non-harmful, can be actually helpful or beneficial. How can the bacteria be beneficial or helpful? Like they can be good for the plants, for example. Like these bacteria in the soil, like in this picture here, they are called nitrogen fixers. Nitrogen fixers. Um, they can take the nitrogen. Nitrogen is one of the components of the proteins. In order to make proteins and other structures, in order to make these structures, you need nitrogen. How can the plants take the nitrogen? Then the nitrogen is extremely high percentage. It's high percentage in the air. But if you look at the plant, their mouth, it's not really mouth, but this is the entrance of their food, is from the roots. And the roots are there underneath in earth. I mean, in the land, inside. So they do not have an access to the nitrogen. How do you get the nitrogen? The bacteria around them can take the nitrogen from the atmosphere, fix it, meaning put it in the form of something in the land, and the roots can take it. The other thing is they can work as also as the decomposers. Um, Decomposers means they decompose. Remember, we talked about those. Some of these bacteria, they live under land, like not just in the atmosphere. They live under the land. So they actually live there, and whatever they find, they can use. They don't need oxygen or anything. They can just work or feed on anything that they can find. In this case, if there is a dead organism in the land or something, or plants or animals or something dead inside, they feed on it and they decompose it. So that when you decompose, D is a prefix that means without. Um, Decomposing, it's composed and they decompose it, means they break it down so that in this case, the plants can take their needs. The plants cannot decompose anything there. Bacteria will decompose it and then they take it ready to use. So this is, of course, the bacteria is not doing that just for the plants, they're doing that for themselves. But as a product of their life cycle or their survival, they also, they are decomposing. So that's going to benefit the plants. What else can be a positive impact or a good thing about the bacteria? Um, they can use it in the sewage treatment. They actually uh, come to these like sludge, and they intentionally they spread these microbes until they decompose whatever they can find there. They know that this is their normal habitat. They breed it, they feed it in the lab, and until it's good enough to be used, and then they put it on sewage for treatment, and then they are going to decompose it and clean it. So this is another thing. The other thing is called bioremediation. Bioremediation. Bioremediation is when you remediate, means you clean up, remove the pollutants from the environment. So they spread these types of good bacteria for bioremediation. These bacteria, they will eat, they will get every pollutant that they can find. So again, all the four that we talked about so far, whether we're talking about nitrogen fixing or decomposition or sewage treatment or bioremediation, bacteria is doing that for themselves. But because they are doing that, why don't we benefit from that? They are doing that anyway. Why don't we benefit from that? They like to eat the pollutants. So if we get it there, they will eat the pollutant. They are good, and it's in our best interest. So why not? We can use it. So this is the first type, which is the helpful bacteria. The other type is pathogenic or pathogens. Patho means disease, translation. Gen means generators or generation. Pathogen means generation of disease. So pathogens are the disease generators. These are some types of bacteria that can cause disease, like any one of those, Salmonella, Staphylococcus, Streptococcus. I mean, you don't have to know the details, but just know that they can cause disease when they come into to us, humans or animals or plants. They can produce disease. If you're talking about humans, we call it pathogens because they can generate a disease. Path means disease. Um, Now, which one is more ancient? Which one is older? Which one was there billions of years ago? These are the prokaryotes. So the prokaryotes were there first, and then eukaryotes came later, billions of years later. So billions of years ago, there was only prokaryotes on Earth. And then later on, about 2 billion years ago, eukaryotes evolved from the prokaryotes. Like prokaryotes become more advanced, and like they started to put this genetic material inside the nucleus and become bigger and multiply and become multiple cells together and so on. So they evolved from the prokaryotes. What's unique about eukaryotes? Most important thing that I mentioned several times is that membrane surrounding the genetic material. And we call the system in general that's unique for this is endomembrane, a membrane inside which things are located. So this endomembrane system is two components, interconnected internal membranes that are internal connected, and other membranes that, like this membrane here, contain this organelle. Like we will discuss the details. Like this membrane contain the mitochondria. This membrane contain lysosomes, for example, and different organisms within the cells. So if you think about it, how did that happen? This is the prokaryotes. Look, DNA, genetic material. Remember, we said the basics are genetic material, cytoplasm, plasma membrane, plus, if we're talking about prokaryotes, plus a wall. But now, how did these ancestor prokaryotes become eukaryotes? Here is what they did. I'm not talking about the cell wall now. They lose the cell wall. Now you have the cell membrane. The cell membrane was told to fold in, to make folds inside, until it ended up making membranes around different organelles. So did these have organelles? Yes, they had some organelles. Prokaryotes have some organelles, not including the nucleus. The nucleus is an organelle. Yes, it's the largest organelle. Organelle means any organ, small organ inside the nucleus. So I'm sorry, inside the cell. So nucleus is an organelle, like a small organ inside the cells. Mitochondria is an organelle. Golgi apparatus is an organelle, and the plasmic reticulum is an organelle. Lysosomes an organelle. So any one of those inside the cytoplasm, inside the cells, is called organelle. So they have it, but they do not have the big organelle, which is the nucleus. How did you do it? They become more advanced. They evolved, and they used part of their membrane to make nucleus, which is surrounded by a double membrane. They make some more foldings to make something like endoplasmic reticulum. An endoplasmic reticulum, rule of the thumb in biology and A&P later, is to understand the origin of every word. So endo means inside. Plasma, referring to the cytoplasm. Reticulum means network. So this is a network inside the cytoplasm. What is this network? It's the membrane folding itself inside to make this endoplasmic reticulum. So now it acquired this endomembrane system to become more advanced and to become eukaryotes. So this is a eukaryote. How did you know it's eukaryote? Because it have the endomembrane system. Where is it? Here. You see this? The genetic material is not like this. This is just genetic material in the cytoplasm. That's it. But here, you see a membrane surrounding it. Actually, it's a double membrane with openings. This is classic nucleus. So now it is eukaryote, not pro. Here is pro. You don't have a nucleus. Here is normal nucleus, plus other organelles that came from folding of the membrane. One of those of the organelles is the mitochondria and the chloroplasts. How did they get that? This is called endosymbiosis or endosymbiosis. Endosymbiosis is one species live inside another species, and in this case, when it live inside the other species, then it become part of it. Like in this case, the mitochondria or the chloroplast, it was not part of this, but it evolved later on. This species got into the inside of this evolving prokaryotes, and when it get in there, it's not there to harm anything. It will survive inside, and now actually it will become part of it, like the chloroplasts and like the mitochondria. By the way, chloroplast and mitochondria, both of those are the energy producers. Um, Chloroplasts mainly in plants, but in this case, some bacteria also have it. And mitochondria is for anything else, like for animals, humans. We have the mitochondria. This is the energy factory. This is where we make our energy. Mitochondria in humans and animals, and other organisms, of course. Chloroplast in plants, and few other organisms. Now, the next part is the eukaryotes. Eukaryotes, I think you're familiar with it now. Eukaryotes evolved from prokaryotes by making that endomembrane system. The membrane folds inside, starts to surround the genetic material, making nucleus with a double wall and pores, and other parts of the membrane folding make like the endoplasmic reticulum. And then an organelle or organism got inside a bigger organism and become part of it. And remember, we call that endosymbiosis. So what are the the domains coming from eukaryotes that have these criteria that I talked about? Animal, plants, and fungi. Remember that we count towards the domain of animals. Whether we agree on that or not, but this is biology. So eukaryotes, plants, animals, fungus. There is an additional one that's called protists. Less advanced, but this is something that we need to know. What is it? They say, okay, so these are plants, thousands of plants, thousands of animals, thousands of fungi. And then they found something that fits into that category of being eukaryotes, but it's not one of those. It is not animal, it's not plant, it's not fungus. What should we call it? So they just give it a name of protists. Protists. So protist is others. You consider it others. Other eukaryotes. What do you mean? I mean anything that's not one of the three. It's not animal, it's not plant, it's not fungus. What else? But does it fulfill the criteria of being eukaryotes? Yes. So let's just call them anything else. We call it protist instead of saying others or other eukaryotes. Just call it protist. So protist is a category. These protists evolved like 2 billion years ago. Most of them are unicellular, but some are multicellular. So listen to this. Remember when we said that the prokaryotes are all unicellular? Eukaryotes, most of them are multicellular. But here is an example. How about the protist? Doesn't the protist count toward the eukaryotes? Yes. Can the protist be monocellular, like one cell by itself? It can. And it can be multicellular. Yes, everything else is multicellular, I understand. But since even one of them, one of the eukaryotes, can be unicellular, so it's safe, it is correct to say that the vast majority of the eukaryotes is multicellular, but some of them can be unicellular. Example, the unicellular protists. But remember, protist does count toward the eukaryotes. It's not one of the main three, but it does count. What types of protists do we need to know? Which is, again, it's a eukaryote. Like protozoa, for example. Protozoa is a protist that they eat. They obtain the nutrients by eating, usually by eating from surroundings. They can invade us, they can invade animals, and but anyway, they don't have to invade. They can just survive like in the water, and they can the bottom line is they eat their nutrients somehow, whether it is from humans, from animals, from plants, from none of those. It can be just from the water, any one of those. And here are example of the protozoa. Paramecium is one of them. And paramecium, it can just live in the water. And in microbiology, this is one of the things that you get some water and they look under the water. If it's not clean, it will contain these paramecium. So this is usually in the water. Giardia, this is one type of the protozoa, which is one type of the protist, which is a eukaryote. And this is intestinal or intestinal parasite. What does it mean? It means it's a parasite that can live in our intestine or the animal's intestine. They live in there. It's parasite. It's not useful. It just take our nutrients, steal our nutrients. So we call it a parasite. If it is eating but also benefiting us, we don't call it parasite. But this is just parasite. Parasite means selfish. They're just there to take our nutrients, and you get sick, that's fine. Plasmodium is another thing, and it's also do the same thing, but Plasmodium, they most likely they live inside our blood cells, like Plasmodium malaria, one of type of the Plasmodium, and this one lives in our cells, in our blood cells, specifically red blood cell. They eat everything in there until the cell rupture, and then they go to another cell, enter inside, they take the nutrients from the cell, and so on. So these are different types of protozoa: Giardia, Plasmodium, Paramecium, and these are all different types of protozoa, which is a protist, which is a eukaryote. What else? Amoeba is one cell type of protist. Remember, protists can be unicellular, can be multi. Uni means one, multi means many, more than one in general. So it can be unicellular, like amoeba. If I ask you, can the eukaryotes be monocellular usually, or unicellular? Usually say unicellular, like animals, humans, plants, one cell? No, actually yes, because you didn't think about the protist. Protist does count toward the eukaryotes. Example here is amoeba. Amoeba is one cell, but it's a protist and it's eukaryote. Others like the slime molds, this is also another type of protist. It looks like a fungus. It looks like it, and their lifestyle is like it. It's not a fungus. It's not a fungus. It counts toward the protist. What else is considered protist? Algae. Algae is plural. The singular is alga. And this is, remember in the beginning, I said that these different organisms, they can take oxygen and produce carbon dioxide like us, plus nutrients, of course. They can do the opposite, like plants, photosynthesis. They take the opposite, carbon dioxide from the air, water, and they take the energy from sunlight, which is the most important. That's why we call it photosynthesis. They take their energy from the sunlight energy. So, but we all know that plants do that. But do you know that there are actually some protists do that, like algae? Yes, algae. Some of them will, and that's why actually they look like green in color, like the seaweeds, for example. This is multicellular, and if you look at it, a lot of people think that these are plants when they see it. Seaweeds, yeah, this is a plant. It's not a plant. It's not a plant. It's an algae. And algae means it lives with photosynthesis in the water. Yes, but they still do photosynthesis, means they take their energy from sunlight. They can make their own, so they don't feed on other organisms, for example. So algae can be unicellular and can be multicellular. Here is another example of a eukaryote that's unicellular. The vast majority are multicellular, but here is the second example. Unicellular green algae. It's green. By the way, anything that's photosynthesis, they have to be green. Why? Because this is the color of the chloroplast, and the chloroplast is the energy generator from the sun. Since you have it, you will be green colored. So even algae can be unicellular like this, and it can be multicellular like the seaweeds. Remember when we said that the protists can be unicellular and can be multicellular as well, like amoeba, like protozoa, and so on. Algae can be unicellular and multicellular. So this is another type of protist that's a eukaryote. Now, when we come to multicellular, remember so far, we're still talking about eukaryotes. What are eukaryotes? Plants, animals, fungus, and others. Others. What do you call others? Protists. And we give some examples like protozoa, algae, and so on. So now the multicellular, which is other things. So the multicellular, the biologist said that this multicellular is another evolution. Remember when the prokaryotes that live all by themselves billions of years ago, and they started to evolve and become eukaryotes by creating a membrane around the genetic material, making the nucleus and other membrane folding, like making the endoplasmic reticulum, and other organisms get into there, live in there, survive, become part of it, like the mitochondria. But all of this evolution is you're just changing from prokaryotes, one cell, unicellular prokaryotes, to unicellular eukaryotes. How can you become multicellular? The unicellular eukaryotes evolved and started to multiply and stay together um, to become multicellular. Multi obviously means many cells. Multicellular. Um, the biologists said that the first 2 million years, there was only unicellular. Unicellular. Multicellular started to evolve after those first 2 billions of years. So, according to the biologists, according to the scientists, Earth evolved about 45 uh, millions of years ago. It's actually billions. Remember that 1,000 million is a billion. So this is the origin of Earth. It's billions, obviously. It's 4,600 million years. So it's 4.6 billions. This is where Earth originated. And then later, which is 3,500 or 3.5 billions, prokaryotes started to appear. Um, and then the eukaryotes started to appear about 2.1 billion years ago. This is where the eukaryotes started to appear as an evolution of the prokaryotes. And then from there, about 1.2 billion or 1,200 million years ago. This is where the multicellular life. So prokaryotes alone become unicellular eukaryotes, billions of years, and about 1 billion, and then later on the unicellular become multicellular, and this is about 1.2 billion. This is where the multicellular started, and then about 600 million, not a billion anymore, years ago, this is where the animals uh, evolved. And then the colonization was about 490 million years ago, and humans started only 2.5 million. Not only, it's a lot, but compared to Earth, this is not much. But anyway, it's 2.5 million years ago. This is where humans appeared on Earth. After all of that is there already. So it's colonized with, you have animals, you have plants, you have fungi, all of those developed. And the last thing to develop is humans. So now, characteristics of lives. This is very important. Characteristics of lives is what should we have in order to say this is alive, this something is alive. You have to fulfill the following criteria or characteristics. Reproduction, you have to be able to reproduce, grow, develop, use energy, you have to have orders, you have to have cells or be a cell. So the question is, virus, is that alive or not? There is a debate about that. It's not 100% of the scientists agree, still debating on it, because it fulfill a lot of the criteria, but not 100%, so it's debatable. So is the bacteria alive or not? Let's go back to the criteria. Does it reproduce? Does it grow? Does it use energy? Does it have an order? Is it in the form of cells? It is not in the form of cells. So the virus has no cell. Cell is what we talked about from the beginning of the chapter. Okay, cell consists of genetic material, cytoplasm, cell membrane. Do we have this here? No, we do not. Does the virus reproduce? Listen to this. On its own. So here again, reproduction is not just reproduction. Reproduction on your own. Reproduce on your own. So like fire, does the fire produce? Yes, it does actually. It's grow and become bigger, right? It produce another fire here. No. This is not it. On your own, on your own. Like if I get this virus and leave him alone, can it become 2, 4? Actually, no. So 2 of those criteria, 1, 2, 3, 4, 5, 6 of the 6 criteria is not there. So that's why the biologists, most of them said, no, it is not. There are 2 criteria that's not fulfilled. It's not a cell, or show me where is the cytoplasm, where is the cell membrane, where is it? But there is genetic material here. Yes, that's not enough. But somebody will argue and say, no, wait a minute. The virus reproduce. Otherwise, when they infect us, why do we get sick? Because it's reproduce. It actually, one virus become 2, 4, and so on, and they move from one cell to the other, causing disease. So they reproduce. No, this is not on their own. How do they reproduce? They go to the body, they go inside the cells. They have to be inside the cells. That's why they call it, we call the virus obligate intracellular. Intra means inside. So they have to be inside the cells. And still, they order the nucleus of the cells, the genetic material, the DNA of the cells, they take over, occupy, and they go to the nucleus and tell the nucleus, make copy of me, because I can't make copy by myself. So does it reproduce on their own? No. But They reproduce, yes. This is where the debate came from. But those who do not consider virus alive, they say, no, it's not just reproduce. We have to make it reproduce on their own. And this is where the debate came from. So what's a virus then? The virus is nucleic acid, and this is the genetic material. They have the genes, nucleic acid inside. Some of them will be nucleic acid, of course, can be DNA or RNA. NA is nucleic acid. Is it DNA or RNA? Both of them are nucleic acids. So some viruses will have DNA and some viruses will have RNA. In both cases, it's nucleic acid, it's genes anyway. It's genetic material anyway. So some of them will have DNA, some of them will have RNA. DNA, deoxyribonucleic acids. RNA is ribonucleic acids. Deoxyribo what? Nucleic acid or ribonucleic acid. In both cases, it is nucleic acid. So the nucleic acid is the main thing. DNA or RNA, it's a nucleic acid anyway. Some of them are DNA and some of them are RNA. Outside of that, you'll have something called capsid. Capsid is an outer coat that's made of different types of proteins, different types of protein molecules surrounding, protecting the genetic material inside. And outside of that, they have recognition spikes, these spikes here, so that they will be recognized. Like if they go to our cells will look at these spikes and they will recognize that this is a virus, and they recognize what kind of virus, so that they can produce their antibodies and they can fight against it. So, but this is how they are recognized, how to know that this is a virus, spikes. So again, nucleic acid as a genetic material, DNA or RNA, surrounded by capsid, and capsid is a protein surrounding the nucleic acid, different types of proteins. And then you have the spikes outside for recognition. This is a virus. There is something here that's important also to know. Everything here is important as long as I'm discussing the details. Everything here is basically important, but some of them are even more important. So there is something called the bacteriophage or phage. Translation, what's a phage? Phage means eaters. Eaters. Bacteriophage, they eat the bacteria by invading them. So these are viruses that invade the bacteria and eat from them, from inside of them. They are called bacteriophage. They infect the bacteria. So bacteriophage is a virus that's phage to the bacteria. They invade into the bacteria. They live inside the bacteria. They eat from inside of the bacteria. And their life cycle can be one out of the two, either a lytic cycle or a lysogenic cycle. And this is a very important topic. Lytic cycle is when the viral DNA, the DNA of the virus, they replicate into the, they go into the cells, they use the cell machinery, and here we mean the nucleus. They order the nucleus to make copies of them. This is how they replicate. And then when they like reproduce more, replicate more and more and more until they culminate enough to fill and wipe off the nutrients of that cell, exhausting the cells until the cell will actually end up dying. And we call that the lytic cycle. So they leave the cells only when the cell is exhausted enough to death. Lysogenic cycle, on the other hand, they do the same thing, but instead of just ordering the cell's machinery, which is the nucleus, not necessarily the nucleus, by the way. I mean, I'm sorry. I mean the genetic material. Whereas it's almost the same. Just the nucleus is more advanced. Genetic material inside the membrane, we call it the nucleus. Genetic material without a membrane. This is the machinery anyway. So on this type, in this case, the bacteria, the machinery of the bacteria, which is basically the DNA of these bacteria. In the lytic case, they are using the machinery and they culminate inside the cells until the cells die. And the lysogenic, on the other hand, they do not do the same thing. Instead, they go and merge with the bacterial chromosomes. Instead of ordering the chromosomes, they go to the chromosomes, order them. This is the lytic cycle. The lysogenic cycle is, instead of doing that, they actually go merge and insert into the chromosomes of the bacteria and just stay like this for a long time. During that long time, of course, the chromosomes of the bacteria are going to replicate. They are going to replicate too because they put themselves inside. So they become part of it temporarily and they can stay there for a long time. So here is the lytic cycle. They invade, use, abuse the bacteria. They use their machinery to replicate enough until they wipe off, and the bacteria end up dying. We call that the lytic cycle. Then bacteriophage can go into other bacteria, and instead they are going to merge themselves and insert themselves into the chromosomes of this bacteria. And here it is inside. And the bacteria is going to replicate anyway. They replicate inside of which, instead of ordering and occupying, they merge. And since the bacteria is going to replicate anyway, so automatically they will replicate with the bacterial chromosomes. The bacteria, including their chromosomes, are going to replicate. 1 become 2, become 4, become 8, and so on. The bacteria itself, including the chromosomes inside, they become part of the chromosomes. So they are going to replicate anyway. And then the viral DNA can, at certain point of time, they can stay like this for a long time, but they can detach themselves and leave at certain point of time. And we call that the lysogenic cycle. So understand the difference between lytic, and by the way, lytic is coming from lysis, and lysis means breakdown. So lytic cycle is the cycle that end up breaking down. Lysogenic, on the other hand, is coming from lyso, like merging, inserting. So this is another cycle. You need to know the difference between those two. Virus, we all know that the viruses invade humans, they can invade animals. But guess what? They can also invade even plants. So like we all know the flu virus, the cold virus, the AIDS virus, the COVID virus, and so on, invading humans. Same thing can happen like swine flu for animals and so on. But actually, there are some viruses that can invade the plants, like this tobacco mosaic virus. It invades the tobacco leaves and they invade the plant itself. HIV as the most important example of viruses. This is a deadly virus because the vast majority of people getting that virus, they end up dying. And AIDS is an abbreviation for acquired immune deficiency syndrome. Acquired because it's not natural, you acquire it from outside. Immune deficiency because you will become deficient of immunity, means you don't have enough immunity, and it's a syndrome. Why did you call it a syndrome? What does it mean? Why did you call it a disease? Disease and syndrome are kind of similar, but the difference is disease is one disease. Like you have common colds, and you know what you get with the common colds. Syndrome is multiple organs are affected. Like what happened to me if I get colds? Well, it's the inside of your nose, basically. This is a disease. Pneumonia, bacteria or virus invading the lung. That's it. This is disease. But if you have multi-organ, multi-systems conditions, like something happened to the bone, and another thing happened to the lung, another thing happened to the blood. Together, we call that a syndrome, not just a disease. HIV virus is called a retrovirus. Retrovirus because it is RNA. Remember, viruses, remember the structure, we talked about it. It is a genetic material, which is nucleic acid, whether DNA or RNA. Can be DNA virus, RNA virus. This is an RNA virus. Means the genome or the genetic material or the nucleic acid that's surrounded by capsid and spikes. What kind of genome do you have? What kind of nucleic acid? RNA. The problem with this specific virus and why it's so deadly virus is because very specifically they invade T helper cells. And remember this, T helper cells. What are the T helper cells? Here it is. We discuss the blood. The blood is cells and fluid, right? The fluid is called the plasma. Cells are red blood cells, white blood cells, and platelets, right? White blood cells. There are different types of white blood cells. One of them, you have like monocytes, like the basophils, and so on. Different types, monocytes, neutrophils, basophils, and so on. One of these types of white blood cells these white blood cells is called lymphocytes. So we have monocytes, we have lymphocytes, we have neutrophils, and so on. But we're talking about lymphocytes specifically. Lymphocytes are the ones that's more advanced and give us a very good immunity. We are not discussing the immunity right now, but generally speaking, we have non-specific immunity, which is like your skin and your different things that generally speaking, protecting you against anything in general, but the lymphocytes is a specific one. They are specified in a certain virus, in a certain disease, and certain thing. And T helper cells is one of the types of lymphocytes. It's T and B lymphocytes. So remember this, blood is cells and plasma. Cells, white blood cells, red blood cells, platelets. We're talking about white blood cells now. Different types, including lymphocytes. Lymphocytes is the one that's specific immunity. Means they are specialized in certain disease. These lymphocytes can be B and T lymphocytes. Why did you give it those two names? One of them generated and developed in the bone marrow, B, bone marrow. And the other one is developed in the bone marrow, but activated in the thymus, T. So this is T. Now, this is how specific AIDS is. They leave everything in the body and go to the blood, not even the blood. White blood cells, not just white blood cells, lymphocytes, not just white lymphocytes, T lymphocytes specifically, not even any T lymphocytes, one type, T helper. T helper lymphocytes, they work as helper to the T and the B, both types of lymphocytes. They help them to develop. So when these viruses, AIDS virus, when they invade the T helper cells, killing them, so now you're losing T helper cells after T helper cells after T helper cells. Who is going to help the lymphocytes anymore? Whether T or B, no help. They will not develop. So when you get a disease, you are waiting for the lymphocytes to come and develop and grow and ready to fight. You don't have that leverage or that ability anymore because T helper cells are type of T cells that help both T and B. For them to develop, maturate, become strong enough to fight, they need the T helper cells. You don't have it anymore, you are not going to develop T and B anymore. So anything can invade you. And usually those people will die from anything that's normal human being that are not infected with those, and they have normal T helper cells, will never even have a chance to die from these diseases. They can die from flu. Flu? Yes, they can die from flu. Why? Because they cannot develop their, like anybody, when they get the flu virus, the T helper cells will develop the T and B lymphocytes, and T and B lymphocytes are going to fight and they will get rid of the virus. It takes some time, a week or something, but that's okay. They will end up getting rid of it. But in this case, it doesn't go to the extent of having zero, but it's very small amount remaining of T helper cells. So you don't have enough, you can die from anything. Even the fungus. The fungus, by the way, is something that's very weak, and it does not invade us. It can invade barely the skin. This is how weak it is. Like the athlete foot, tinea, they can invade, like this is fungus, they can invade the skin because they are weak. But if they got inside, they will die. But in this case, they will get infected, they get fungus like inside their lungs. This is extremely rare. In normal people, fungus cannot do that. Like if it started even to go inside, you will wipe them off. They are too weak. But you are even weaker than them if you have AIDS. So they will go in, nothing is facing them. So why not? Let's just invade. And as we know, how the HIV virus or the AIDS virus, how they spread from person to person. We all know sexual interaction, sexual contact, whether it is hetero or homo, whatever it is, it's sexual interaction. Shedding blood products, receiving blood from somebody who's infected or exposed somehow, even with a needle. So it goes into your body through the blood or through the membranes, sexual intercourse or blood products. When we mentioned that, the HIV virus or AIDS virus is an RNA virus, what's the significance of that? What's the importance? Why are we saying that? Because DNA can give orders to the nucleus or to the genetic material to do whatever they want. RNA does not have that leverage. RNA is not as strong, it's not a manager. DNA is like the manager of the cell. RNA is not. So RNA by themselves cannot give orders to the genome, whether we're talking about humans or animals or bacteria or whatever. They invade into the cells, but how can I, as an RNA, how can I give an order to the DNA of the cells? I can't. I can't give, like, DNA is more stronger, and this is the boss. I cannot. So they have something unique in that case, and this is called reverse transcriptase. Transcriptase is an enzyme that helps DNA to make RNA. We call that transcription. Reverse is the opposite. You have RNA from which you make DNA. So the RNA of this virus, using reverse transcriptase enzyme, they become DNA. They produce, convert RNA into DNA, and if that's the case, DNA can order the cells to make more copies of them. And when they fill the cells, they leave, go to another cell, get inside, use the reverse transcriptase, make DNA, convert RNA into DNA, take over the cells, give orders to make copies of them, and so on. So they hijack. By the way, all viruses hijack our cells. They hijack, they invade, they occupy, they give orders to the DNA. The only difference is, if you are a DNA virus, you just give orders to the DNA inside the cells that you invaded. But if you are RNA, you don't have that strength. So what are you going to do? You're going to use that special type of enzyme, reverse transcriptase, To convert your RNA into DNA, now you can give orders. And here is the life cycle of the HIV. And I already discussed it. They come into the human cells. They inject into they inject their genome. In this case, it's an RNA. RNA cannot give orders, so reverse transcriptase to make DNA. DNA is the one that go to the nucleus, go to the DNA of the nucleus, order them to make more copies of their viral DNA, and then it is going to leave, go to another cell, but it also end up going to the T helper cells and occupying them. You're losing T helper cells after T helper cells until it takes years, different from person to person, but it takes like long time until they destroy and occupy a lot generation after generation of the virus until they end up wiping off or destroying the vast majority of your T helper cells, and any disease can kill them at the end. And actually, just for your information, not only that, but because they go like this, they can actually make an issue inside the cells in them ending up producing certain types of cancers. I'm not going to go into the details, just I wanted to tell you that those people will die either from vast majority die from any type of pathogen, virus, bacteria, even fungus can kill them because of their T helper cells are destroyed, and some of them can develop actually cancers because of this sophisticated entrance into the DNA of the cells. And that's it for this chapter.